Morning, everyone. Today now we'll discuss blood physiology. I am Mr. Madhusudan Patidar, and today we will discuss blood physiology. Uh, I am faculty of Index Department of Physiotherapy and Paramedical Sciences. Blood physiology. Blood is a special type of fluid connective tissue derived from mesoderm. The branch of science concerned with the study of blood-forming tissues and the disorder associated with them is called hematology. Hematology means hem means blood and logos means study. Combined hematology, study of blood and blood-forming organs. Now we'll discuss properties of blood. Blood color. Blood color is bright red in artery and dark red in veins. Mass 8% of the body mass. pH. Now we'll discuss pH. Slightly alkaline. pH 7.35 to 7.45. Taste salty. Temperature. Now we'll discuss temperature. 38 degree centigrade and Fahrenheit 100.4 degree Fahrenheit. Now we'll discuss viscosity 3 to 4 times more viscous than water. Now blood is 3 to 4 or 5 times more viscous than water. Volume. Now we'll discuss volume. Volume is 5 to 6 liter in adult person. The healthy adult person is 5 to 6 liter volume of total blood under the body. Composition of blood. Now we'll discuss the plasma part. There are 55% of plasma and 45% of hormone element like RBC, WBC, platelets. Plasma, yellow color complex and hormone element RBC, WBC, platelets. Blood. There are composition of blood. There are two types of defined plasma, 55% plasma, and the cellular part, cellular elements of 45% of cellular elements. Under the cellular elements, RBCs or erythrocytes, WBC or leukocytes, platelets or thrombocytes. Plasma. Now we'll be discuss plasma. Definition of plasma. Plasma is a pale yellow colored liquid component of blood that holds the cellular elements of blood in suspension. Plasma is a pale yellow color liquid component of blood and 55% of present in the under the blood. Total blood volume of 45% is cellular part and 55% of plasma part under the blood. Constituents of plasma. Now will be discussed constituents of plasma. Plasma. There are water part of 91%, 91 to 92% of water part, and proteins. 7% of protein present in plasma. Like albumins, globulins, fibrinogens, there are plasma proteins. Other solutes. 1.5% of other solutes under the present in plasma. Other solutes like electrolytes, ions, sodium, potassium, calcium, magnesium, chloride, HCCO3, and sulfates. Gases. Now we'll be discuss gases present in under the plasma. Other solutes. Gases O2, CO2, N2, oxygen, carbon dioxide, and nitrogen. Now we'll be discuss nutrients present under the plasma: glucose, amino acids, fatty acids, etc. And some waste products present in the plasma, other solutes: urea, uric acid, ammonia, creatine, etc. Now we'll be discuss functions of plasma. Constituent water. Functions: water absorbs, transports, and releases heat. Albumins osmotic balance, globulins defense mechanism, fibrinogen blood clotting. There are mostly fibrinogen is important work in blood clotting. Electrolytic ions, pH buffering. There is some functions of plasma and important functions of plasma. Now we'll be discuss red blood cells or erythrocytes, yep, or RBC. Shape. Now we'll discuss shape of RBC: circular, biconcave, non-nucleated shape in RBC. Size. Diameter 7 to 8 micrometer. Diameter RBC and thickness 2.5 micrometer in thickness of RBC. Color. Red color. hemoglobin pigment. Under the presence of hemoglobin pigment of color is red. RBC count, normal count of RBC, yeah, or erythrocytes. Adult male 5.5 millions RBC per microliter. Adult female 4.8 millions RBCs per microliter. Now we'll discuss lifespan of RBC. Lifespan of RBC 120 days in normal lifespan of RBCs. Erythropoiesis. The production of RBCs is now as erythropoiesis. The production of RBCs is now as erythropoiesis. Adult red bone marrow of long bones, hip bones, breastbone, and ribs. Child up to 5 years. Bone marrow of all the bones. Fetus, liver, and spleen. Erythropoiesis is a process of formation of blood cells, erythrocytes. That is erythropoiesis. Formations of RBC now will be defined erythropoiesis. Increase in number of RBCs is now as polycythemia. Polycythemia is a disease. Increase in number of RBCs is now as polycythemia, and decrease in number of RBCs is now as erythropenia. There are two types. Polycythemia means increase in number of RBCs now as polycythemia, and decrease of number of RBCs now as erythropenia. Clear? Now we'll be discuss functions of RBCs. RBCs mainly functions of transport O2 from lungs to tissues and transport CO2 from tissues to lungs. It is a mainly functions of RBCs. Normal blood contains 13 to 15 of SB per 100 ml of blood. Uh, one RBC contains about 250 million molecules of SB. It is a now one RBC contains about 250 million molecules of SB hemoglobin. Okay, normal blood contains 13 to 15 of SB per 100 ml of blood. 100 ml of blood, 13 to 15 gram of SB per 100 ml of blood. Each molecules of carries 4 molecules of oxygen and is important work of transport O2 from lungs to tissues and transport CO2 from tissues to lungs. Now we will discuss white blood cells, WBC, are leukocytes, safe amyloid nucleated, safe in WBC, size 12 to 15 micrometer in size. WBC colorless, translucent. WBC is colorless. Count 5,000 to 10,000 WBC per microliter, 4,000 to 11,000 WBC per microliter. Lifespan 10 to 13 days of lifespan WBC in the normal condition. And different types of WBC, white blood cells. There are two types of defined, two types of classified WBC: agranulocytes and granulocytes. Granulocytes, under the granulocytes, neutrophil, basophil, eosinophil, and under the agranulocytes, monocyte, lymphocyte. The types of WBC, different types of WBC, and mainly work of mainly immune system. Uh, Now immune system work. WBC is defense against bacteria and viruses. WBC is leukocytes. Leukopoiesis. Now we'll discuss leukopoiesis. The production of WBC is now as leukopoiesis. Leukopoiesis is the process of formation of WBC from bone marrow to blood stream. Leukopoiesis is the process of formation of WBC via formation of white blood cells from bone marrow to blood stream. The production of WBC now is leukopoiesis. Adult formation of WBC, liver, spleen, tonsils, bone marrow, fetus, liver, and spleen. Sites of formation of WBC. Increase in number of WBC now is leukocytosis, and decrease in number of WBCs is now as leukopenia. Pathological increase in number of WBC now as leukemia, blood cancer. It is a leukemia condition of blood cancer. Pathological increase in number of WBC more than WBC is now as leukemia. It is a blood cancer. Increased number of WBC now is leukocytosis, and decrease in number of WBC is now as leukopenia. Now we'll be discuss types of WBCs. WBCs, there are two types of WBC, granular and agranular. Uh, Now we will discuss granular types of WBC. Neutrophils 62%, acidophils 2.3%, uh, acidophils or eosinophils, and the third types of basophils 0 to 4% in under the WBC. Now we will discuss agranular lymphocytes and monocytes. 30% present lymphocytes under the WBC and 5.3% monocytes present under the WBC. Now there are five types of WBC: neutrophil, eosinophil, basophil, monocyte, and lymphocyte. There are two types of agranular and granular sites. Now we'll be discuss granular WBCs types. Neutrophil. There are features neutrophil nucleus with 3 to 5 lobes. Under the under the neutrophil, nucleus is 3 to 5 lobes. Stain with neutral dye hematoxylin. Stain with neutral dye hematoxylin. Functions: destroy bacteria by phagocytosis. Destroy bacteria by phagocytosis. Location produced bone marrow. Now we will discuss eosinophils. Features: nucleus with two lobes. Under the eosinophils, nucleus with two lobes. Stain with acidic dye with eosin. Stain with acidic dye with eosin. Functions: combat the effect of histamine in allergic reaction. Allergic reaction work is mostly eosinophils. Location produced bone marrow. Now we'll discuss basophil. Nucleus with indistinct lobes, basophil. They are or irregular shape nucleus, distant with basic dye methylene blue. Liberate heparin and histamine in allergic reaction to intensity inflammatory response. Location produced in basophil bone marrow. Now we'll be discuss agranular WBCs. Now will be first lymphocytes. Features: smallest of WBC. Lymphocyte is the smallest WBC. In a large round nucleus under the lymphocyte present in large round nucleus. Functions: functions of lymphocyte produce antibodies. Uh, Location produced bone marrow, skin, tonsils. Now we'll be discuss monocyte. Uh, Monocyte is a large WBC or largest WBC, large kidney shaped nucleus under the monocyte present of large kidney shaped nucleus, engaged microorganism. Functions: location produced in monocyte, bone marrow. Now we'll be discuss platelets. Shape circular, biconvex, non-nucleated in shape size 2 to 4 micrometer. Count 8 lakh to 4 lakh platelets per microliters in normal count of human being. Lifespan of platelet 5 to 9 days of lifespan under normal condition of platelets. Functions: blood clotting. It is an important main function of platelets, blood clotting under the clotting mechanism. Thrombopoiesis. Now we'll be discuss thrombopoiesis. The production of platelets is known as thrombopoiesis. The production of platelets is known as thrombopoiesis. Uh, Bone marrow from blood stem production of platelets. It is defined as a thrombopoiesis. Platelets are the fragments of large cells called megakaryocytes that remain in the bone marrow. Increase in number of platelets is known as thrombocytosis. Increase in number of platelets is known as thrombocytosis. And decrease in number of platelets is known as thrombocytopenia. There are two types: thrombocytosis and thrombocytopenia. Increase of number of platelets is now thrombocytosis, and decrease in number of platelets is known as thrombocytopenia. Now we'll be discuss blood clotting. Blood clotting is the process in which blood loses its fluidity and becomes a jelly-like mass few minutes after it is set out. Process of blood clotting. First, blood vessel is punctured. Second, platelet from a plug. Third, uh, prothrombin activator. Prothrombin Ca2 plus present soluble fibrinogen and present in thrombin Ca2 plus insoluble fibrin. Third, platelets and damaged tissue shall release prothrombin activator, which entices a cassette of enzymatic reaction. Fourth, fibrin third formed and trap red blood cells. And now will be damaged tissue is injured tissue is a fibrin third release of fibrin third from trap red blood cells and plug is complete. Now we'll be discuss anticoagulant. A substance which prevents the coagulation of blood is called as anticoagulant. A substances which prevents the coagulation of blood is called as anticoagulant. Heparin is a natural anticoagulant present in the blood. Heparin is a one type of natural anticoagulant present in blood and under the body of human. If blood clots too easily, the result can be thrombosis. Clotting is an under damaged blood vessels. If blood clots too easily, the result can be thrombosis. Clotting is an under damaged blood vessels. If the blood takes too long to clot, hemorrhage can occur. Thank you. I am Mr. Madhushan Patidar, faculty of Index Paramedical College. Thank you.